All right, looks like we are recording. So, hello everyone. Welcome to the Thomas Expert Series. Thank you for joining us today. We're going to get started in just a moment. In the meantime, please double check, make sure your microphones are muted. This expert series will be recorded and placed on demand within 24 hours. So please let your colleagues know how much you enjoy the presentation today and direct them to the on-demand tab or have them sign up for future talks. To get everyone a little familiar with the chat, I want to see where everyone's tuning in from today. I'm in New Jersey. Don, where are you at?
I'm in Pennsylvania.
Okay, so not too far. Let's see what we've got in the chat. We've got South Dakota, North Carolina, Florida, Jersey, Texas, cool. Virginia, Maryland, Indiana, Connecticut, cool. All right, at this time, I'm going to go ahead and turn it over to Don here to talk about the impact of Annex 1 and your contamination control strategy.
Okay, thanks, Heather. Hello, everybody. It's afternoon, morning, wherever you are in the U.S. Like to welcome you. This presentation is going to be about Annex 1 and the contamination control strategy. So I'm going to discuss the latest Annex 1 revisions and the relevance to cleaning and disinfection practices, the significance of closed processes within this field, as well as what a contamination control strategy is meant to be. Please note that the presentation was put together and directed for your customers' point of view, which means they're pharma and biopharma and cell therapy manufacturers. Okay. Now, over a 5-year period of time, the regulators from the PICS organization, which is the Pharmacopeal Inspection group, which includes the FDA and EMEA, plus over 50 other countries, they wrote and rewrote a series of drafts which have some major and minor changes to Annex 1 guideline, which is basically the good manufacturing practices for sterile medicinal manufacturing. The final version of this revision process added clarification for the use of quality management as recommended from ICH Q9 and Q10 guidelines. Okay, so let's first look at what was desired to be the significant changes. The scope and the principles were updated.
I don't know what his draft process was, like how much he actually wrote.
In the scope, it now states that design and control across the manufacturing facilities and systems should incorporate the principles of quality management, basically risk management. The intent always has been to ensure control of microbial, particulate, and endotoxin contamination prevention. Now, as an added statement to the scope, it includes that any non-sterile manufacturing products, processes, or parameters are allowed to follow some of these same principles. But if it's determined appropriate by a manufacturer to follow some of these, then they must document which of them are being applied. Now, the basic principles have not changed. Thus, the GMPs include design, qualification, and validation of facilities, equipment, and processes. Then we have training and qualification of personnel. Monitoring systems should be designed, qualified, and then used for monitoring purposes, but also they should be monitored as well. And then, of course, excipients and raw materials should be controlled to be fit for use. Overall, not only quality risk management, but quality management is key. This means having and improving control of people and the manufacturing processes. So now the newest terminology, although not new to microbiologists and quality personnel, is contamination control strategy. Now, the strategy is meant to be developed by subject matter experts and management together. Yet the importance of the strategy is how the strategy will be met. This strategy has been part of multiple revisions of Annex 1 over the past 5 years, so it isn't new to anyone who's been following these change recommendations. As of August 2022, those who need to follow the Annex 1 guidance are expected to have a strategy in place, a plan to meet the strategy in a format that can be presented to an inspector, and then also confidently supported as being followed. Now, I'm going to provide a brief overview of what we've learned about this control strategy and where we're expected to be when an inspector comes to our facilities, and then what can be the next steps for us. So we've learned that the term holistic is related to understanding that systems are integrated, and we must look at them in this manner. We're becoming better at performing risk assessment to determine gaps and finding mitigating actions for contamination prevention. We are ongoing learners about quality management and how significant it is to our inspectors and reviewers, especially the FDA, and that the whole process must be ongoing as a continuous improvement activity and thinking to assure that we keep up with current knowledge and technology. Now, to build this picture of what a contamination control strategy means, we start with some foundational building blocks. Now, each of these requires input and discussion by a team of subject matter experts. We learn scientific knowledge, technical knowledge, process knowledge as relevant to our process and product. We also learn and become better at assessing risk regularly to determine where the gaps occur and what to do about them. Now, what we learn, we must communicate across the organization as a continuous improvement quality culture. Now, what we look to assess risk is everything that matters in these broad categories of systems: facilities, utilities, materials and waste, personnel movement, personnel hygiene and gloving and gowning, and of course, the raw materials, including actives. Once we've identified the risks, we must determine or communicate the controls that are or will be in place. These controls should be reviewed periodically as a routine, along when an issue develops. And then wherever we can validate, we should do this. If not validate, then an appropriate qualification should be performed. Now, as with any GMP validation, periodic revalidation is expected to prevent aging from causing an issue. And as you can see, validation crosses all these categories where control is key, and it ranges from the QC lab to facility engineering, all the way to the manufacturing personnel and process. Of course, this would include media fills or process simulations, if you know them as that, for aseptic processes. Now, monitoring is also an expectation, not just a wish. Across all these categories, if you don't monitor, then you don't know if something changes. Monitoring is basically taking a measurement where you want to know if a system is changing from what is expected. Now, communications is an important part in an organization. For manufacturing, what's going on is important to upper management, just as what strategies are meant to the production line. Problems need to be escalated, and oversight is important, especially to ensure adequate resources are available, as well as adequate budget and management support when and where needed, all the time. Now, the House of Contamination Control, which you just saw, as I've described, also needs a feedback mechanism. And that feedback mechanism has to do with the continuous mechanism of feedback called change control and CAPAs, which are basically the indicators of mechanisms which allow you to carry actions when necessary and to keep the process running safely for the ultimate customers, which are the patients of the pharmaceutical industry. Now, that brings us to the first question. Where should we be now with regards to Annex 1? Well, with our team of subject matter experts having experienced the risk assessment process, we're expected to be able to show that a team exists, that they're trained in risk assessment, and that there is a champion of that team named. We should also be able to show that a plan for gap assessment has been documented and that most of the risk assessments were performed. The completed mitigations or controls for high risks should be understood and either put in place already, or CAPAs should be in place documenting when this will occur and in an urgent manner. Now, as I mentioned, the same as we know for good manufacturing practices, all risk assessments, all controls, the contamination control strategy and its program should be documented. Any plan for controls and timings that are not implemented yet should be documented when that's going to occur. So once a manufacturer's subject matter expert team has performed the contamination control strategy and the risk assessment activities, the next steps would include training the rest of key personnel in order to develop a quality culture and understanding what contamination control is. Now, there should be a plan in place for who will face the inspector in order to ensure that that person is fully knowledgeable of the contamination control strategy. And the contamination control strategy, or CCS, as stated, must be an ongoing process with periodic reviews scheduled and documented when it does occur. Now, part of continuous improvement with the CCS and culture is ongoing learning. The management must support this across the organization. As inspectors ask Annex 1 type questions, the operators will learn what they're looking for, and the inspectors will learn how well operations will mature in this area. All inspectors will follow Annex 1, whether it's PICS, EMEA, or Health Canada format. They'll be investigating against these guidelines. Again, they expect manufacturers to have a CCS document and implementation of that strategy in place, which includes all the risk assessments and a plan or schedule for periodic review. Now, manufacturers are expected to also have ongoing monitoring in place to meet the contamination control strategy. They're expected to have the appropriate validation or qualification where applicable, and they're expected to show communications with governance occur as part of the organization understanding of Annex 1. Now, let's take a closer look at what some of these key clarifications that have now occurred in the new revised Annex 1. Starting off, cleaning and disinfection activities, it's now important to understand and show that there is a cleaning step which is followed by the disinfection step. In other words, cleaning is an important preliminary step to disinfection. And since residues or soiling can present a physical barrier to disinfectants or hydrogen peroxide vapor if used, the presence of residues can bind or interfere with disinfectant actives. And if residues are not removed, then disinfection may not be that robust. Excuse me. Now, there's always been an ongoing question about what disinfectants to use or rotate, and now it's a bit clearer by Annex 1. Here it states that more than one disinfectant is expected, and it probably should be a sporicide. So in other words, you have a broad spectrum disinfectant, for instance, and a sporicide. Now, disinfectants which are used in the grade C and D areas, which may not be considered to have to be sterile, have to be identified on whether they're going to be sterile or not. But if there's any formulation preparation needed for some of these concentrates that may be used in the non-sterile areas, in-house prep may require some sterile components and may be required to be aseptic. And if you decide to use it sterilely, then you have to have WFI testing, filters in place, filter integrity testing, QC testing, and validation to show that you've been able to convert a non-sterile product into a sterile product to use it in the grade C or D. But if you decide to use non-sterile, then it needs to be documented, and then you can cover and justify that piece. Either way, if you decide to use a ready-to-use formula, which is already sterile, then a certificate of analysis has been stated now to be acceptable for these ready-to-use products, as well as the validation documents that back up the ready-to-use products. So that also has to go with vendor qualification. So you can't just use documents unless you've qualified the vendor to support that. Now, specifically stated now is that validating the disinfection process includes data from actual use conditions, along with data from different surface materials. Now, what does that mean? It means that there probably needs to be some laboratory testing with different surface materials, and then also some in situ testing in the facility after cleaning, performing environmental monitoring in order to validate that the way that the disinfectants have been applied, that they're going to work, and you've got data to back it up. In addition, in-use expiry has also been stated to be important, and it needs to be developed using validation data that the user is able to generate themselves or with help from the suppliers. Now, quite common today, in a number of instances, modern facilities have automated decontamination using hydrogen peroxide vapor. Now, Annex 1 actually states that this can be used, but it needs to be validated. Now, another piece is the idea of closed processes, mainly for aseptic processing, but obviously it's very commonly used in the cell and gene therapy industry. But anywhere from the QC lab to manufacturing use, barrier technologies such as isolators or RABs in some cases are expected, and only those two are expected. The movement of materials also, or products, into and out of these barriers must be robust, proceduralized, and validated to ensure contamination is not carried from the lower classified environment to the higher classified environment. Now, as transferring items, as I mentioned, from a low classification to a high classification without sterilization can lead to contamination, then this transfer process is expected to be validated to show that the prevention in place is robust. So if you're transferring through a pass-through hatch, for example, it can be more controlled if it's designed appropriately. So one way of designing it appropriately up front is to have an automated process built in that will help make the materials and their prevention of contamination more robust and allow for hydrogen peroxide vapor decontamination in that pass-through hatch. Now, one requirement that hasn't changed, but it's a very important requirement, and that's training. Training of employees in all the appropriate areas of knowledge are very specifically stated now in Annex 1. It doesn't just say training appropriate for the job, it actually states now in training in microbiology and hygiene, in clean room practices, in aseptic techniques. It's now stated in Annex 1 as an expectation of the inspectors. So just to give you a little bit of background, if you haven't heard it before, Ecolab has a group of global technical consultants which can assist you in supporting your customers and whatever their needs and technical requirements are. So we're a resource for you guys to utilize anytime you need it. And now we can tie all this up in a summary in a nice package of understanding some of these new requirements about contamination control as it's stated in Annex 1. So manufacturers are expected to have a CCS in place and a high-risk area mitigations in place and implemented. There's a growing concern for removing residues. The disinfection program must be in place. And if you have to prepare any sterile disinfectants from non-sterile ingredients, it needs to be balanced by the appropriate processes, procedures, and validation, or the use of ready-to-use formats, which already have some of that documentation from the supplier in place. Validation of disinfection itself must show justification in the way that it's applied to the surfaces in the facility. And Ecolab continues to recommend control measures that are aligned with Annex 1, and our technical support team is ready to support you and provide timely access to our expertise whenever you and your customer need it. Any questions?
Thanks, Don. While you guys are typing out your questions, I do want to take just a moment and share my screen with you. Ecolab does have literature as well as web links and videos within their kiosk, so please take a look around and view some of their great resources. And there will also be the on-demand talk within 24 hours. So if you go ahead and click on this, you could see all of our on-demand talks. And this one will be up within 24 hours. So I will go ahead and hand it back, and hopefully there are some questions in the chat.
Okay, now let me open up the chat. Okay, let me roll back and see. Okay, first question. Annex 1 has always been related to sterile manufacturing. So how do you determine when or where to use it for non-sterile manufacturing, as you mentioned earlier? Okay, so the question has to be, since Annex 1 has always been related to sterile manufacturing, it's now stating that you can use it for non-sterile manufacturing areas as long as you state why and where you're using it to the inspectors. So this says, when or where can you use it in non-sterile? Well, basically, if you think about this, a lot of non-sterile manufacturers. Now, the easiest one is pharmaceutical manufacturers, even the oral solid dosage manufacturers, or those that make topical creams and ointments. They quite often have their formulation and their filling operations in a clean room type room. They may or may not classify that room as a clean room. They may have it classified just for particulates, or they may have it not classified at all. But still, it's a clean type room. And because it's a clean type room, the intent is to keep it clean so contamination does not find its way crosswise into the product, whether by the operators, materials, or just in the air. So in that case, it doesn't hurt to have a program in place, a good disinfection program that can follow the appropriate needs of those surfaces that need to be disinfected. Thus, a validation of those materials, which is following what Annex 1 is requesting, is not a bad idea. As long as you can put that together and you're confident that it proves that what you're using is what you're intending to get out as an outcome. I hope that answers the question. Let's try next question. Is rotation of more than one disinfectant a GMP requirement? Oh, that's a really good question. I'll tell you this. Rotation, as the term rotation has not been used. It's been put in and taken out, put in and taken out, a lot of drafts of a lot of different guidance documents. I haven't seen a regulatory document yet that actually has it in there, but I'm sure there's some across the regions of the globe. It's found its way in there. But the basic problem with the word rotation is that it's saying that you need to rotate, but not how often. And people think that rotation means every other time you use something, you've got to change it. And the problem in disinfection is that if you don't understand the chemical nature of your disinfectants, then you could be how do I put it? You could be putting one disinfectant down and then the next one can be reacting against that disinfectant and neutralizing it. Now, that could be a good thing, but on the other end, it may not be a good thing at all if you're intending to disinfect with that second reagent. Now, the other piece is that people are, like in Annex 1, using a sporicide in the quote rotation, even though it doesn't state that it has to be a rotation. It's saying use at least a second disinfectant. Using a sporicide is a good idea if you've recovered bacterial spore. Disinfectant. Now, you got to be very careful when you're recommending to people that want to use these, especially the sporicides, in that they need to use the sporicide sparingly. It doesn't need to be in there every other time. It's almost like use your broad spectrum disinfectant, which will do a good job as long as your environmental monitoring tells you and you understand what's there. It will do a good job disinfecting on a regular basis. And then maybe once a month or once every couple of months, you go in and use a sporicide and then rinse that sporicide off and then move back to your normal schedule. Because if you try to use a sporicide on a every other basis, you're going to end up being very wary that you may be leaving residues of the sporicide. And sporicides are probably the strongest oxidizers of all the disinfectants. And a strong oxidizer means it's going to cause damage to a lot of different surfaces. It can lead to corrosion. And most of these sporicides also have a very strong odor that people who are applying them do not like to be in the midst of for a long period of time. So the idea of rotation, the word itself may not have shown up in there, but you have to understand that they're saying use more than one disinfectant to make sure that you understand what your environmental flora is, what you're recovering, and that you're going to be killing all those flora. It is possible that using one disinfectant is enough. But that's why in the case of Annex 1, if somebody comes in for that type of inspection, you can say, I'm using this broad spectrum on a regular basis, but once every so often, I'm putting in my second disinfectant, which is the sporicide of what they expect. Okay, I hope that it's a long-winded, but rotation's been a really big question that's always shown up in disinfectant efficacy questions. If GMPs require disinfectant validation, who is expected to perform this validation, the user or the supplier? Okay, that itself is a good question because there used to be an expectation that all of the data would be supported by the supplier. Or if you're very conservative in a big pharmaceutical company, you're going to say, I don't trust the supplier. It's conflict of interest. I'm going to have all those studies done internally by my lab, or I'm going to contract them to a contract lab. The GMPs do not require anybody in particular to perform those validation studies, especially the laboratory studies. But it's recommended, especially by us, since we're a supplier, that a third party perform those. And quite often, some of the suppliers now are contracting third parties to perform these tests in the laboratory. Now, the requirement for the in-house testing or validation, which means you perform cleaning and disinfection, and then you follow it up with environmental monitoring. That probably should be done by the user themselves. And I'm only saying that it depends on resources. They may have a contract lab group come in and perform their environmental monitoring. And they may have a third-party contract group doing the cleaning and disinfection, but it all needs to be coordinated appropriately so that it is performed one way or the other. And then they can deal with the data and then be able to determine, do they have to worry about conflict of interest or not? And probably not if you're dealing with the third parties, and that should be able to follow suit. Okay, on that question? Let's see, do we have any other questions here?
I'm not seeing anything. Anyone have any other questions that they would like to ask? And you can unmute yourself if you don't want to type it out.
And I don't know whether my email was shared with anyone, but Heather, I give you permission to share my email with anyone. If you ever have any questions to be able to ask that your customers have asked you that you'd like to have a little bit more background information to help you out, please feel free to contact me.
Awesome. Thank you, Don. If no one else has any questions, I will go ahead and close us out. Thank you all for joining us today, and a special thanks to Don for joining us. And I hope you all have a great rest of your day.
Thank you all.
Thank
Bye-bye.
you.
Thanks, Don.
You're welcome.
Sound